Cell division plays an important role in life. In terms of prokaryotic and unicellular eukaryotic cells, the division of one cell reproduces an entire organism. Cell division also enables multicellular eukaryotic cells to develop from a single fertilized egg. After such an organism is fully grown, cell division continues to function in the renewal and repair, replacing cells that die from normal wear and tear or accidents. The trillions of cells that undergo biological processes in the human body depend on efficient cell division. Additionally, regulations and checkpoints throughout the division process prevents defects in the cell, such as uncontrolled cell growth. Cells do most of their growing during G1. In addition to cell growth, the cell will begin synthesizing producing proteins, cytoplasmic organelles, and enzymes related to DNA replication. During the S phase, chromosomes are replicated and DNA synthesis takes place. During G2, organelles and molecules required for cell division are produced, and the cell is ready to enter mitosis. The mitotic phase consists of mitosis, the division of genetic material in the nucleus, and cytokinesis, the division of the cytoplasm. Mitosis is conventionally broken down into 5 phases. During prophase, the chromatin fibers become tightly coiled, condensing into 2 identical sister chromatids joined at the centromeres. The nucleolus will disappear, and then the mitotic spindle fiber will begin to form. The centrosomes will move far away from each other, propelled partly by the lengthening of microtubules between them. The cell will now enter prometaphase. During this stage, the nuclear membrane will fragment and the chromosomes have become even more condensed. The microtubules extended from the centrosomes invade the nuclear area. The 2 chromatids of each chromosome develop a kinetochore, a specialized protein structure at the centromere. On the other hand, the non-kinetochore microtubules interact with those from opposite poles of the spindle. Now, in the metaphase stage, the centrosomes are now at opposite poles of the cell. The centromeres of the chromosomes line up on the metaphase plate. During anaphase, the cohesion proteins are now cleaved, which allows the 2 sister chromatids of each pair to part suddenly. The 2 daughter chromosomes begin moving toward the opposite ends of the cell as their kinetochore microtubules shorten. The cell elongates as the non-kinetochore microtubules lengthen. During telophase, the final phase of mitosis, 2 daughter nuclei form in the cell, and the nuclear envelope begins to arise from fragments. The nucleolus will reappear, and the chromosomes become less condensed, and all remaining microtubules depolarize. Cytokinesis, or the division of the cytoplasm, varies greatly among plant and animal cells. In animal cells, cytokinesis occurs by a process known as cleavage. The cleavage furrow, a shallow groove in the cell, will appear. On the cytoplasmic side of the furrow, a contractile ring will contract and pinch inward, forming 2 completely separated cells. In plant cells, vesicles from the Golgi apparatus move along microtubules toward the center of the cell. The vesicles will coalesce, forming a cell plate. The cell plate will grow until its surrounding membrane fuses with the plasma membrane, resulting in 2 daughter cells with a cell plate between them.
There are 3 checkpoints during the cell cycle that allow the cell to divide properly and thereby prevent defects in the cycle such as uncontrolled growth of abnormal cells. The first checkpoint that must be completed is the G1 checkpoint. The G1 checkpoint determines whether all conditions are favorable for the checkpoint with the correct size, the minimum level of energy it requires, and there is no DNA damage. A cell that does not pass these tests will not go on to the S phase. The cell can then stop the cycle and attempt to go into G0 phase, also known as the inactive phase, and await further signals when conditions improve. It will progress to the G2 phase where the second checkpoint is. At this checkpoint, the cell will recheck the cell size and protein reserves, but most importantly, it will check to make sure that all of the chromosomes have been accurately replicated without mistakes or damage. The M checkpoint occurs near the end of the metaphase stage of mitosis. The M checkpoint checks to make sure the sister chromatids are correctly attached to the spindle microtubules For cell cycle regulation, there are external and internal signals that tell the cell when to divide. External signals include physical signals, which include cell-to-cell contact called density-dependent inhibition. An example of this is how mammal cells can be grown in a culture dish, and they then form a single layer of cells on the bottom and then stop dividing, which is an example of the physical external cell. Another type of external signal is a chemical signal. The growth factor is platelet-derived growth factor, or PDGF, and PDGF is required for division of cultured fibroblasts. The fibroblasts each have a tyrosine kinase called PDGF receptor. Once PDGF is bound to this receptor, it triggers a signal transduction pathway that allows for the cell to pass the G1 checkpoint and therefore divide. For the checkpoints, an internal signal would be at the M phase checkpoint. There is a signal that happens in the cell that delays the beginning of anaphase if the spindle fibers are not properly connected due to the nidochore not being properly connected to the microtubules. Once the spindle fibers are properly attached, a signal is sent and the regulatory protein complex and begin anaphase. There are mainly proteins of two types, protein kinase and cyclins. Protein kinases phosphorylate other proteins to activate or inactivate them, which allow for the G1 and G2 checkpoints to be passed. So these kinases are called cyclin-dependent kinases, or CDK. The activity of a CDK increases and decreases depending on the amount of cyclins available, which is all how the G1 and G2 checkpoints can be passed. The G2 checkpoint can also be passed using the maturation promoting factor, or MPF, whose activity corresponds to the peaks of cyclin concentration. Cyclins accumulate during G2 and bind with CDK molecules, which result in a MPF complex, and that MPF complex phosphorylates a variety of proteins, initiating mitosis. An example of cells that do not leave the G0 phase are heart cells. This is due to the fact that once a heart reaches full maturity during your adult years, it is no longer producing cells to grow and therefore do not divide again. In normal cells, hundreds of genes intricately control the process of cell division. Normal growth requires a balance between the activity of those genes that promote cell proliferation and those that suppress it. Cells become cancerous after mutations accumulate in the various genes that control cell proliferation. Over time, these cells become increasingly resistant to the controls that maintain normal tissue, and as a result, they divide more rapidly than their predecessors. Scientists hypothesize that the cancer cell does not stop dividing in the absence of a growth factor, like a normal cell, because it either is making its own growth factor or there is an abnormality in the signaling pathway that allows cells to believe it is receiving a growth factor. Either way, as we already learned, the external signal of growth factors are important to pass the G1 checkpoint, but since there is no stopping the growth factor, the G1 checkpoint fails and cancer cells continue to multiply at an insane rate.